So here are some electron micrographs of biofilms. You can see here the microorganisms embedded in the 3D gelatinous matrix. In different ways you can see that they have formed microcolonies inside the 3D gelatinous matrix. What is the microbial biofilm actually made up of? Let us now see in the coming slides the composition of the microbial biofilm. The microbial biofilm consists of a 3D extracellular matrix in which the bacteria are embedded. The gooey matrix that sticks bacteria to each other and to the surface is composed of a complex array of substances that are grouped as extrapolymeric substances. This EPS is a highly hydrated gel-like biopolymer that immobilizes the bacteria. In general, the EPS consists of exopolysaccharides, extracellular nucleic acids like extracellular DNA, extracellular RNA. It consists of proteins, lipids and other biomolecules. One should remember, however, that the composition and the structure of the extrapolymeric substance can vary greatly depending on the type of microorganisms, the shear stress, the availability of the nutrients and the host environment. The extrapolymeric substance of different biofilms shows a considerable molecular, structural and functional diversity. Most of these components are made up by the microbial population. Sometimes biomolecules acquired from the host or the surrounding environment may also be present in the biofilm matrix. Lately, therefore, a new term has been coined to encompass this diversity in microbial biofilms, and this term is matrixome. Here is a schematic representation of the main components of the biofilm matrix and how they bind the bacteria together. Biomolecules that make the matrixome can be grouped into two major categories: those associated with the cell surface, which you can see here in the form of molecules like adhesins, and those which are secreted extracellularly, which you can see in the form of exopolysaccharides, proteins, extracellular DNA, RNA, etc. Those associated with the bacterial cell surface can also be molecules like flagella, the type 4 pili and also some functional amyloids. The 3D extrapolymeric matrix of the microbial biofilm helps in adhesion, and it also gives a scaffolding for the microorganisms to embed themselves into. This 3D matrix also offers mechanical stability and protection to the biofilm. Most of the times, the biofilm 3D matrix is heterogeneous and differs according to the niche it has been formed in. The structure of the biofilm is very interesting. It is not just a blob of extrapolymeric matrix that is attached to a surface. The structure has been now found to be an intricate network which is for nutrient flow to happen within the matrix. It is almost like a circulatory system that develops in the 3D matrix of the biofilm. This structure therefore allows nutrients to flow in and nurture the cells within the biofilm and the toxic end products to flow out of the biofilm. So here is a diagrammatic presentation that shows us the 3D structure of a biofilm. The first one is the bulk biofilm, where you can see how the water channels are formed in the 3D structure. These water channels, as it's shown, allow the flow of nutrients in and out of the biofilms through the biofilm architecture. It also shows us the discontinuity of cohesion, that is the heterogeneity of the biofilm, where you will see some pores and channels formed. It also indicates the heterogeneity in terms of metabolic activity, where one can see that there are gradients of fluid movement as well as oxygen concentrations that diffuse through the biofilm. The second one is showing the internal composition of the biofilm, which shows us how the microorganisms are entangled in the polysaccharides, proteins, extracellular DNA, etc., and how these microorganisms form microcolonies inside the biofilm. After seeing the structure of the biofilm, one also understands that biofilm is not generally uniform but may vary spatially and temporally. Biofilm morphologies can be different, like you can have smooth biofilms, flat biofilms, rough, fluffy or even filamentous biofilms. Biofilms can have a lot of degree of variation in their porosity. Three structural variants of the biofilm matrix have been identified: the heterogeneous mosaic, the porous biofilm that is interdispersed with water channels, and a dense confluent biofilm. These morphologies are influenced by the surface properties, the environmental conditions that are there, the nutrient content available for the biofilm to be formed, which definitely leads to an increase or a decrease in the extrapolymeric substance that is present in the biofilm. It has also been found that when high amounts of EPS is produced, denser biofilms are formed, whereas low nutrient conditions lead to biofilms that are less dense and interdispersed with a lot of water channels. Another interesting phenomena is biofilms formed under high velocity normally give rise to streamers and ripple-like structures as shown in the diagram. Here are some confocal microscope images of microbial biofilms. You can see the 3D structure or the 3D architecture of the biofilms in these pictures. Some of them also seen as top view of mushroom structures that are formed in the biofilm. The other confocal image is showing biofilm that is formed under low nutrient conditions and therefore has a interdispersed structure with many water channels. So far, we have discussed what are biofilms, how they are different from planktonic cells, how they are significant, and what are the advantages that a biofilm have over planktonic cell population, what these organisms have an edge over the planktonic population. We have also discussed what biofilms are made up of and how their 3D architecture is. Let us now move on to understand how the biofilms are formed.